In today's society, vaccines are a common way of preventing many dangerous viruses. In the 1950s, at the height of the polio epidemic, people were in dire need of a vaccine for the paralytic disease. Doctors and researchers were willing to try anything to fix this problem facing millions of Americans. During the early 1950s, researchers tested the polio vaccine, not only on animals, but also on children with physical and mental disabilities. The trial as a whole, however, was well regulated and carried out methodically. The 1954 Salk vaccine field trial was a revolution in vaccine testing procedure. In 1894, the first major polio epidemic in the United States struck 132 citizens in the state of Vermont. By 1916, more than 9,000 cases devastated New York. The turn of the century marked the rise of a new disease that was misunderstood and seemingly uncontrollable. Frightened parents kept their children indoors and away from public places like swimming pools, parks, and schools. Researchers identified three types of polio. The first two types were the most common. They often led to paralysis in the limbs. Type 3 was the most damaging. It infected the brain, leading to paralysis of the diaphragm, which often caused death. Patients with type 3 polio were quarantined in isolation wards and lived up to 3 weeks with an iron lung. In 1935, two physicians, Maurice Brodie and John Colmer, each decided to try their hands at developing a vaccine. Using the spinal cords of polio-infected monkeys, Brodie raced to get his vaccine ready for a frantic society. Using a live virus method, he unwittingly tested his vaccine on humans. His field trials resulted in healthy individuals contracting polio, and in some cases, even death. The subsequent Colmer trials were canceled. President Franklin Delano Roosevelt, a patient of polio himself, would announce the creation of the National Foundation for Infantile Paralysis. The NFIP, better known as the March of Dimes, would later play a major role in the funding for the vaccine.
In this case, it was the American people that paid for that trial through the National Foundation for Infantile Paralysis March of Dimes. Kids in movie theaters and schools and going door to door to houses, people would give dimes, literally, which then ended up being sent to the White House. And it was those dimes, dime upon dime upon dime, that ended up funding all of the research programs themselves and then the conduct of the field trials. So this was not government doing things, it wasn't industry doing things, this was the American people.
In the 1940s, polio cases spiked again. Between 1945 and 1949, the U.S. averaged 20,000 polio cases annually. One important researcher who worked on solving the crisis was Dr. Jonas Salk. Salk had worked with his mentor Dr. Thomas Francis Jr. on the development of the influenza vaccine during World War II. During this time, both doctors developed a deep interest in the killed virus vaccine. The killed virus technique is different from a live virus method because the patient would not be directly receiving an active strain of polio. In 1947, he accepted a research position at the University of Pittsburgh and began work involving the polio virus. The majority of the polio researchers at the time felt that a live, weakened virus vaccine was the only method of preventing polio. But Dr. Salk felt that the safety benefits of a killed virus vaccine would outweigh the effectiveness of the live vaccine. Using the methods developed by Dr. John Enders, Dr. Salk and his lab staff began the rigorous process of developing the virus in the kidney tissues of rhesus monkeys. The vaccine was then killed using a formaldehyde mixture to deactivate the virus. After rigorous testing of various vaccines, Salk was able to create a batch that he considered to be both safe and effective. The Salk vaccine had been created. Before the March of Dimes would consider testing the vaccine on children, Dr. Salk would first have to prove its safety. The vaccine's safety and efficiency was first continuously tested on mice and rhesus monkeys.
You can't do work of this kind and begin to get answers to what may be effective and what won't be effective without using animals. So I think that it's really important that the animals be taken care of well, cared for, not abused.
When the vaccine proved safe in the monkey tests, Dr. Salk moved on to testing his vaccine on children who had already been paralyzed by polio. Salk thought that by testing on children who were already exposed to one of the three possible strains of polio was an obvious start to the human testing process. He began testing at the D.T. Watson Home for Crippled Children, where children with polio were injected with the vaccine and any side effects were monitored and recorded. Dr. Salk moved on to the next stage of testing his vaccine when it failed to infect any of the Watson Home children. His next move was to test at the Polk State School, a home for mentally handicapped children. By today's standards, testing on a mentally handicapped child is morally wrong, but back then they viewed testing on children whose future potential was already limited as a necessary precursor to testing on healthy American children. The Polk school tests ran smoothly. It was time to distribute the vaccine. With polio at the forefront of many Americans' minds, the field trial was sure to become a widely publicized event. In order to ensure the trial's neutrality, the foundation hired Dr. Thomas Francis Jr., famous for developing the influenza vaccine, to run the trial. This revolutionary move would ensure the trial was separated from the politics of the foundation. Dr. Francis accepted the challenge and set up a vaccine evaluation lab at the University of Michigan. Dr. Francis's first major contribution to the trials was to modify the layout. Instead of the vaccine trial being simply observational, Francis insisted that the trial be composed of two smaller studies, one of which would be observational and the other a double-blind study. 33 states participated in the observational study, in which first, second, and third graders were observed in order for the year's polio trends to be recorded. 11 other states participated in the double-blind study.
The trials started out small in the Pittsburgh area, got bigger in a special school, and then in the local Pittsburgh schools. And then, when it came time really to go out with a big field trial, my father did not want to have it be a placebo-controlled trial. As it turned out, it was an enormous field trial, the largest ever conducted and probably the largest ever to be conducted, involving 1.8 million school children, not all of whom got the vaccine. Some got the vaccine, some got a placebo, and others were just part of the experiment as so-called observed controls. But for those kids that got placebo, in other words, it wasn't real. It was salt water or something of the sort. My father just couldn't stand that idea because they could have actually gotten vaccine, and he just didn't want to have kids go unprotected if they could have been protected.
In addition to the 1.8 million school children involved, 20,000 physicians, 40,000 nurses, and over 200,000 volunteers helped make the trial possible. Over 237 gallons of the vaccine were used. In the rare event of a death of a participant, Dr. Francis would personally evaluate the cause in order to ensure the safety of the vaccine. On April 12, 1955, the 10th anniversary of FDR's death, Dr. Francis stood in front of the crowded Rackham Lecture Hall at the University of Michigan and announced that the double-blind study demonstrated that the vaccine was 68% effective against type 1, 100% effective against type 2, and 92% effective against type 3. The Salk field trial occurred during a time when unethical human testing was common. The notorious Tuskegee syphilis experiment was underway during the development and the testing of the Salk vaccine, and the infamous Nazi human experimentation had occurred only a decade before. HeLa cells taken from a poor tobacco farmer without our knowledge were becoming an essential tool in medical research, and the team who conducted the tests at the Polk State School considered them to be humane and well-regulated. In the 1950s, researchers believed that the ends justified the means. Although the tests at the Polk State School were considered controversial by today's standards, they were humane and well-regulated. The large field trial was proof that a breakthrough in ethical and safe human testing was possible during a time when it was not the norm. Although the methodical nature of the Salk vaccine field trial would not be continued in the majority of medical trials during the 1950s and 60s, it would leave behind a legacy of how a major trial could be properly run. Even though the trial was considered by many to be the riskiest public health experiment ever, it revolutionized safety measures and proved how cohesion and unity in creating a large organization led to success that changed how medical research was funded. Even though some of the measures that went into making the trials possible were controversial, such as animal testing and using mentally handicapped children as early test subjects, the trials still served as a basis for forming today's medical ethics. The trials united and helped American citizens to fight against the debilitating effects of polio and revolutionized how we think about medical ethics today.